Okay. Hello everyone, welcome to PAPERI webinar series. I'm Rahma from Egypt. I will be today's moderator. Thank you for joining us for the session today. I'm delighted to introduce our esteemed speaker, Dr. Hugues Abriel. Hugues Abriel has degrees in life science from ETH Zurich 1989 and human medicine from the University of Lausanne 1994. He has managed research and teaching departments and served on the board of Swiss National Science Foundation's presidency. Since 2022, he has been Vice-Rector for the Research and Innovation and a member of the Executive Board of the University of Bern. Since 2014, he has maintained regular contact with scholars from the African continent, particularly from the Democratic Republic of the Congo, Morocco, and more recently Senegal. Building equitable relationships with African colleagues has become a priority of his career, and he has consistently sought to make global academic communities fairer and more respectful and more inclusive for everyone. His research focuses on channelopathy, rare human diseases caused by ion channel dysfunctions, such as cystic fibrosis, cardiac arrhythmias, and epilepsies. His group is also assessing the feasibility of nanopore-based DNA sequencing for genes implicated in rare and monogenic diseases, particularly in African populations. In today's session, Dr. Hugues Abriel will talk about democratizing genetic diagnosis, nanopore-based CFTR sequencing across the African continent. Cystic fibrosis remains largely underdiagnosed across Africa, and access to precision therapies for affecting patients is severely limited. In this talk, he will present the work of Dr. Nada El Mahden and colleagues at the University of Fez, Morocco, whose recently published studies in Frontiers in Genetics 2026 demonstrates the visibility of long-read nanopore sequencing for CFTR variants discovered in Moroccan patients, including the identification of multiple novel variants and variants of uncertain significance. He will then discuss how this works fits with the broad framework of Pan-Africa CF research, which he co-founded with Dr. Marco Zamboli, which aimed to build a continent-wide research infrastructure for CF diagnosis, genomics, and ultra-access to target therapies for African patients. Please join me in welcoming Dr. Hugues Abriel. Please note that we will have a live Q&A session at the end of the webinar. You can write your questions and have the access to submit it. Please mute your microphones. Also, these sessions will be recorded on our YouTube channel. Thank you so much and welcome, Dr. Hugues. The floor is yours.
Well, dear colleagues from Cairo, from Egypt, thanks a lot, dear Wafa, dear Rahma. It's a pleasure for me to be able to present in front of this audience the work we have been doing on that specific gene and specific disorders. And actually, you said already a lot about what I'm going to present in this introduction. So what are channelopathies? Channelopathies are diseases, human diseases that are caused either because there are genetic modifications in the genes coding for the ion channels or regulatory proteins, or as you know, here those are the different ion channels that are depicted here, for instance, a calcium channel, sodium channels, and potassium channels. And there are also so-called acquired channelopathies. I mean, if you take a drug, for instance, a drug that would be a neuroleptic, it may also block ion channels in the heart and lead to cardiac problems and arrhythmias. We have been working on genetic channelopathies since about 30 years. And we have been mainly working with cardiac and neurological channelopathies. And here, just to show you, if you block a potassium channel here in the heart, so you can get this kind of arrhythmic event. This is a so-called torsade de pointe in a patient that has taken a drug blocking the potassium channel. There are like 50, 60, 80 types of channelopathies that have been described in the past. And in fact, here what I'm going to tell you is about a personal story journey that I started years ago when I started to be in contact with African colleagues. And it started in 2014, 2015, when I had my first visit to University of Kinshasa. And here you see my two young colleagues at that time, but now they are 10 years older, Professor Jerry Mubungu and Dr. Claude Kayembe, with whom we have been working since many years together. And Professor Mubungu and Dr. Kayembe, they are both pediatric doctors at Kinshasa. And when we are talking about ion channel disorders, so among the disorders that were, we addressed was cystic fibrosis, and you will hear about that in more details in a minute. So when we're talking about ion channel disorder channelopathies in African populations, I mean, very, very little is known. What do we know about genomic and genetics in Africa? So you know it, Africa is the most genetically diverse region in the world, right? I mean, you see here all these different colors actually illustrating this diversity. It has to do with the human history, right? I mean, Africa is the cradle of humanity. And in Europe and the rest of the world, so the populations came out of Africa about 50 to 60,000 years ago. And in fact, if you compare to the rest of the world, each African human genome has about 25 more polymorphisms than each non-African genome. So they are more viable, and that's very important to take into account when we want to do precision medicine. Despite this, my good colleague from South Africa, Professor Shaida Musa from Stellenbosch, when she started a program on genomics, she heard sometimes, genomics is not a priority for Africa. We have other problems. And I'm sure that you understand that we all think that this is completely wrong. So that's a first introduction. And now, since I've been working with ion channels since many, many, many years, so I was following this development of this idea to sequence DNA molecules using nanopores, and has been mainly a company that has done that, the Oxford Nanopore technology. And what they did, they worked on protein nanopores. Those are actually proteins that are coming from different types of bacteria. And in fact, these nanopores are large enough that DNA, single-stranded molecule of DNA, can go through the nanopore. In fact, they have been, devising a very sophisticated concept that by using a motor protein that they call motor protein, which is an helicase, you can unwind DNA molecules, push these DNA molecules across the A nanopore, and then record the current that you can get between the different, sides here. And then by doing so, you can sequence, you're looking at the current, you can get a sequence of the bases here. Obviously, it's much more complex than that, but this is the basic principle. And if you do it, if you multiplex this on a nanopore device, a sensor array, so you can have 2,000 of these nanopores, and you can, in parallel, sequence hundreds and hundreds of molecules of DNA. By applying or implementing this concept, they were able to have small devices, and one of them is here. This is the called Oxford Nanopore technology MK1C device. This is portable, and I was clearly very intrigued by those kind of devices. So what is interesting here? Yes, it is portable. It is quite cheap. I mean, about 5,000 euros to buy the whole device. And then you can, instead of other technology or compared to other technologies, you can get full or very long read, like more than 100,000 of bases that can be sequenced. Usually it's more like a bit from 6 to 10,000 that we are sequencing. And by doing so, it's much easier to look at structural variants in the genome, and for some disease, this is very advantageous. So that's the nanopore technology. Now the question, how can we apply this nanopore technology for specific disorders, and in particular channelopathies? And here the first channelopathy we were interested in the lab is cystic fibrosis. What is cystic fibrosis? Cystic fibrosis is a multisystemic disorder, a recessive inherited disorder, where we have two pathogenic variants in the so-called CFTR gene, and this CFTR gene is coding for an ion channels, actually a chloride channels or bicarbonate channels. And because this ion channel is expressed in many, many different organs and tissues, so kids that are born with two pathogenic variants have severe problems. Gastrointestinal problem, they have failure to thrive, and then most of the time there are also big problems with the lungs, because this channel is important for the clearance in the bronchus, and then they get infected in the lungs and they will die very young, mainly because of severe infections of the lung. So, but you can see here that there are many, many other symptoms that that can be observed with these mutations in the CFTR gene. So the gene is encoded in chromosome 7. It's a large gene with 27 exons, and the whole length of the gene is about 250 kbs, 250,000 bases. And then the protein is a protein that is coding for an ion channel. I mentioned that, about 160 kT and a bit more than almost 1,500 amino acids. So this is a membrane protein, and this membrane protein is an ion channel that is able to let chloride and bicarbonate ion going through the cell membrane. What do we know about epidemiology of cystic fibrosis? Cystic fibrosis has been described since many, many, many years, but now, since about 30 years, we know exactly what are the genes, and this has been described mainly in the white populations in Europe and as well as in North America. And then there, in Europe and North America, now as of today, about 99 to 95% of the patients that are born, and here those are numbers, so we think that there are about 2,000 kids that are born with cystic fibrosis in Europe and in North America. Most of these kids are diagnosed because there are also newborn screening tests. But in fact, what we don't know, or we know less, is that we can find cystic fibrosis in every continent, South America, Africa, and here India. And when we try to make estimates, those are really rough estimates, it is very likely that there are many more kids that are born with this disease in South America, in Africa, and India per year. The big problem here is that many, specifically here in Africa, many of these kids are not diagnosed, and they will die very early, maybe before age 5 or 10, because of severe complications of the disease. So there is a huge gap in terms of taking care of these kids and diagnosing this disease. And this is one thing that I thought a few years ago that I would like to try to address. So what is known about cystic fibrosis on the African continent? Well, first of all, cystic fibrosis affects 1 out of 2,000 individuals in European descent. This is actually a very frequent genetic disorder in the white population. And there is one specific variant that's very often found. This is a deletion of 3 bases. This is the so-called delta phenylalanine 508 mutations that you see here. So a few epidemiological studies have been made in South and North Africa. And for instance, in South Africa, it is estimated that about 1 out of 12,000 South Africans in the mixed ancestry populations would have cystic fibrosis. And there has been a review looking at all the different variants that were found on the African continent. This is quite an old review now, 12 years review, and we would have to do it again. But you can see here that most of the variants were found mainly in all the North African countries, as well as in South Africa. But there is a huge gap of knowledge in all the different sub-Saharan countries. And again, as I said, this is something that we really would like to address. So among the symptoms of cystic fibrosis is that there is a problem with sweat. Actually, sweat is enriched, or it has way too much sodium chloride. The sweat is salty. This was one way to diagnose the patients. And here you see, for instance, on the skin of a patient with cystic fibrosis, these are salt and NaCl crystals on the skin of that patient. And in fact, one way to look at or to diagnose the disease is to perform a sweat test to look at the concentration of chloride in sweat. And there are instruments for that. And about 3 years ago, we launched a crowdfunding campaign in Switzerland to buy a sweat test device that we wanted to share with our colleagues in Morocco. It's about 10,000 euros. And then this crowdfunding campaign went extremely well. We got much more than what we expected. So we could, in fact, buy 2 of these sweat test devices. We brought one to Fez. And here it's a picture of a medical student that worked with us. And this was the first time we performed this sweat test recording in a young patient in Fez Hospital in Morocco. But the other one was sent to our good colleagues in Kinshasa. So now we have sort of a working flow where you can go from a possible diagnostics in terms of you have signs and symptoms, suspicion of CF, and we can get a sweat test. And when you have the sweat test that is positive, then the idea is really to get a genetic diagnostics. And I'll show you how we will do that and how we perform a new way to look at the complete CFTR gene sequencing. And indeed, this has been mentioned in the introduction. This has been the work of a PhD student in the lab, Nada Marsen from Fez, who just got her PhD at the University of Bern. And this is a demonstration that we can perform a complete sequencing of the gene and get variants that are causing CFTR. So I mentioned CFTR gene is a very large gene, and you can see here the gene, 27 exons here, chromosome 7. And the approach that we tried is to cover the whole gene with amplicons, so PCR amplify this whole gene with long-range PCRs. So the idea was initially to make 25,000 bases long amplicons, and then if you do 10 times 25, you got to the whole gene, 250 kb, and then indeed we were able to amplify all the whole gene. Actually, we needed one more amplicon. We did that with 11 amplicons. And then we sequence it with the machine that I just showed you, the MK1C. And you can see here, first, some of the sequence are, a snapshot of a part of the gene that has been sequenced. Here, a better representation. This is a view of the amplicon 5. And here you can see, for instance, that, well, we have a very high coverage. But you can see here that here there's a clear deletion of 3 bases. But we have these deletions that is about 50% of the time. So it means that these patients is heterozygote for exactly the very well-known so-called delta F08 mutation. And this patient had another mutation in trans, sorry. And that with these patients, we were able to figure out what were the causative variants for that disease. Another example here, this is a case from Fez, Morocco, a young patient with respiratory problems, blocks of airway. We performed a sweat test. The chloride concentrations was really high, about 113 millimolar per liter. It should not be more than 80 millimolar per liter. It had all other problems with the pancreas. And here again, we found two variants with the approach that I just mentioned. One variant that is coding or missense variants where we have serine, proline instead of serine in 364, and here an early stop codon in the arginine 1162. Here we have pedigrees of this first study that we performed. Those are 6 different pedigrees from the hospital of Fez in Morocco. And 3 of the probants, they had a positive sweat test. You can see here in green. And in these patients, we had, with the approach that we just mentioned, in the first pedigree, we were able to figure out the 2 pathogenic variants. They were known to be pathogenic because they were in database of pathogenic variants. And here we found that variant that were also CF causing, in that case also. But we also found a variant that was non-pathogenic. And maybe more important here, in these 2 patients where the sweat test was positive, we found new variants. It was not completely unexpected because those are, again, new populations that were not really investigated in the past. So we had this serine 364 proline and another here phenylalanine 1078 serine. Those 2 variants were not found in databases, but since these variants are in trends, they clearly could be pathogenic variants. So at that stage, we call them, VUS, variants of unknown significance. But in our lab, we are able to look at the expression of these different variants. And here, I'd like just to remind you that a membrane protein is, once it's synthesized, has to be folded. Most of the membrane proteins, or many, are post-translationally modified with glycosylation, and it happens in the Golgi apparatus. And then you have the membrane protein with here the sugars. The mutants, and here this is one of the mutant, again, this is the F508 deletion mutant. So these mutants are not well folded, so we call this a misfolding protein. It will be, in fact, recognized by a quality control mechanism of these folding proteins, and it will be degraded. That's why there will be very little and no mature CFTR protein at the cell membrane. One can quite easily look at this maturation process by looking at the Western blot of cells expressing wild type and cell expressing mutant channels. So when you look at cell expressing wild type channels and you have specific antibodies, so you see 3 bands: chloroglycosylated band, complex glycosylated bands, and the full mature band. This is this band that is, in fact, the one at the cell membrane that is functioning for transport of chloride or bicarbonate. And in general, we don't find such third band, or we call it also C band, when there is a mutation like the F508 mutation as shown here. So, and this is also interesting because the new treatment that we have for CF are quite specific, actually, for these different molecular alterations and cellular alterations. There are new drugs that are given in combinations, and some of these new drugs are called correctors because they can rescue the folding. And one of these corrector molecules is called lumacaftor. While there is a drug that may work as a potentiator. It means that if the protein is at a cell surface and then with a potentiator, it may work better despite the fact that it is mutated. So we really wanted to know about these 2 variants of unknown significance that we found in these patients from these small cohort in Morocco, in Fez. so the SER-Pro and the phenylalanine serine variants. And what we did, we expressed in HEC293 cells these variants, and we looked with a Western blot, and you can see here the Western blot of a normal CFTR protein. We have these 3 bands that I just mentioned before. Now, if you look at the positive control, and again here, there is no mature band here, it means that there is a misfolding, there is a problem with the trafficking, and you just have the 2 bands of the non-mature CFTR protein. And we were actually pleased to observe that the 2 variants that were thought are VUS, they also have this maturation problem. There is no 3rd band. And because there is no 3rd band, so it means that those are pathogenic variants. Now, we can classify them or, promote them from VUS to pathogenic variants. That's what we observed here. Then what we could do also with these different cells expressing the variants, the mutated and the wild-type channel, we can treat these cells with the potentiators and corrector drugs. And then this is a treat therapy of ivacaftor, tezacaftor, and elexacaftor. And if you treat these cells for about 24 hours with these 3 drugs, then you see indeed that for the delta F08, you can get a rescue of the mature band. I mean, this explains why these patients with that specific variant 508 can increase or do much better under treatment. What we observed is also that one of these 2 variants was responsive, even better than the F108. Well, that's this phenylalanine 1078 serine, and you see here that we can really very well rescue the mature band. This was not the case for the serine to proline variants, and we would suggest or expect that this patient carrying these variants, unless he has another variant, would not be rescued with this drug. So you can see here that we can go from clinical symptoms, we can go through diagnostics, genetic diagnostics, and get some sort of clue about pharmacotherapy in these patients. This is work that has been expanded by Professor Bedreddin El Nassen in Morocco. Actually, Bedreddin is the older brother of Nada. He's a medical geneticist there. And you see here with the MK1C device. And so now they expanded this cohort to 36 patients. This is a manuscript in preparation. We have more variants. We are more VUS. Among the variants that we have observed, some of them are eligible for the different corrector and potentiator drugs. Some are not. I'm not going to address this aspect here, but this is, again, really, when we are talking about precision medicine, this is exactly what one can do in CF patients. So those are the different variants at the table that summarizes this. I'm now mentioning another aspect of this project, because when I was presenting this to colleagues in African genetics meetings, so I met Professor Rokhaya Ndiaye from Senegal, and she told me, oh, we're extremely interested because in Dakar, we also think that we have CF patients. And in January this year, I was able to bring, because there was a very generous donator from Switzerland, I was able to bring one of these nanodoc devices to perform this. And here you see the different pediatric doctors, Professor Ba, who is the head of neurology at the Children's Hospital of Dakar, Professor Indou Deme, who is the head of genetics in Dakar. And in fact, a couple of days after bringing this device there, I got a WhatsApp call from Professor Ba, and he told me, oh, we had our first positive sweat test yesterday. I asked, very interesting, what are the symptoms and what is the age? 12 years old male, bronchiectasis, no digestive symptoms, and sweat test conductivity above 86, which is clearly pathogenic or it would lead to it's a diagnosis for CF. Now we are still doing they will do the genetic test for that patient. So I'm going to conclude to mention a few projects that we still have with our colleagues. And again, because I told you, while we know a lot already about cystic fibrosis in North Africa and South Africa, there is a lot to be done in many, many different countries in sub-Saharan Africa. So with a neurologist from South Africa, from Cape Town, Professor Zampoli, and now other colleagues, we created the Pan-African Cystic Fibrosis Research Network. And now we have about 14 countries and people, scientists from 14 countries that are members of this Pan-African Cystic Fibrosis Research Network. A few pictures of our colleagues. Actually, we are many more than that. I think we are now about 60. And the goal of this research network is to advance equitable diagnosis, care, and research so that every person living with CF in Africa is seen, diagnosed, supported, and included. So one aspect that we're working on is to work on a Pan-African, if possible, registry of CF patients. There is a South Africa registry. There is a registry in Egypt, but in other countries there is nothing. And the idea is really, to take the example of the European registry, the South African registry, and to have a Pan-African registry. We'll start with many different countries initially. We start with Egypt, Tunisia, Ethiopia, Morocco, and Libya. And really, the dream is that in about 5 to 10 years, we'll have 54 countries that will be part of this Pan-African registry. Another thing that we are extremely pleased, the Swiss National Science Foundation has been funding a research project that we call First Steps in Advancing Cystic Fibrosis Research in Africa. And again, this is with these partners that we have in Morocco, in Congo, but also in Nairobi, Kenya. And then for 4 years, this is going to be very exploratory. All these centers, they have a sweat testing device. We will include patients that are suspected with cystic fibrosis. We will sequence the complete CFTR gene by using the approach that I just mentioned. And so this will increase our understanding of CFTR variants in these African populations. So I will conclude it now. So I think, this audience should not be convinced that, or that is convinced that medical genetics and genomics on the African frontier is what we call, the new frontiers, for many good reasons. For many other reasons, cystic fibrosis as a monogenic disorder, recessive disorder, is a paralytic disease in the sense that, it's clearly illustrates how well, when we can do it, precision medicine is useful for the patients. Again, I don't know in which country you are now, but cystic fibrosis is present in many African countries. And then we are, with our Pan-African CF network, we're convinced that we'll play, a key role for the African people with CF. I'd like to thank you here. Those are all my collaborators in many different African countries. The collaborators of the Pan-African CF Research Network. Again, a big thank you to my colleagues, Marco Zampoli, Leah Ratner, Muriel Hensmer at University of Bern, Sharon Bordieu, Sheila and Narij in Libya, Abate in Ethiopia. Thanks a lot. I'm going to stop the sharing, and I'm really looking forward to hear your questions and comment that you may have.
Thank you,
Yes.
Dr. Hugues Abriel, for this insightful presentation. As someone interested in research, it was so insightful for me and so inspiring. So thank you so much. We will open now the floor for questions from our audience. If you have any question, please use the raise hand feature or type your question in the chat. I will be addressing it. Okay, Dr. Wafat here. Okay, doctor, you have the floor.
Thank you, Dr. Abriel, for your insightful presentation. You said that you make or open collaboration with countries like Egypt and others to build registry for cystic fibrosis, right?
That's correct, yes.
Okay, so um, in your registry, do you depend on just one in reporting on in this registry? You depend on specific hospitals, or you want to depend on specific governorate for cancer? What I know in cancer, for example, when we report in Egypt. What I think there is a Gharbiya governorate that reported cancer incidents. So, what's the plan you follow to report registries, specific hospitals or specific governorate inside Egypt?
Well, the strategy that we started with is to work with people that already have sort of a database, right? I mean, and again, in Egypt, there are hundreds and hundreds of patients, and there are at least two centers. I think one at Sham University and one at Cairo University that are following patients. And the idea is really to start first to have the different centers in the same country to work together, right? And we have the same problem in Morocco, because in Morocco we have Casablanca, we have Tangier, we have Rabat. So, but in fact it's such a sort of a coalition of the willings. Anyone that want to participate, that have patients, even if they have two patients, they can submit their patients in our registry. We have a limited number of variables in the registry so that it's easy. So, for instance, yes, I mean, a few symptoms, whether the sweat test was positive or negative. Actually, we expect it is positive. Whether a genetic test has been done, and then we will build this registry, slowly, so that we can get as many centers and countries with us.
And
We
from
start.
this, Mhm.
We started with South Africa and Egypt because that's where we have the most patients so far.
So from this point, you encourage more and more the motivation behind this to become a member in this network
Sure,
first.
sure.
Yes, and inside or this center will encourage more centers inside Egypt to develop database and to that allow them to collaborate with you
Right,
and to become a member in the network.
right.
Perfect. Is the next plan or step in this network to build a biobank for cystic fibrosis in particular to help patients with cystic fibrosis?
Well, obviously, so one of the things is that the treatment that we have now, I mean, that I mentioned, that are extremely efficient treatments, right? I mean, they have completely revolutionized the treatment, the care of these patients. The expectation is that people with cystic fibrosis that are treated with these three drugs, and maybe other versions of these drugs, will live as long as everyone, right? I mean, the life expectancy will be completely normal. That's the hope. The problem is that as of now, in the US, in Europe, this treatment costs about more than $200,000. More than 200,000. It's extremely expensive. Most of the time, you cannot afford this in countries of Africa. So the hope is that with a well used or well-formed registry, we can convince these companies to work with us and to give the drugs to these patients, because they know that we will be taking care of these patients. We are following them, and that they will again, be able to have maybe in the future a new market for these drugs.
Thank you so much. I will leave others to ask questions to answer them. Thank you.
Thank you, Dr. Wafa. Here in Q&A, we have, could this nanopore workflow be adapted as a generalizable model for other underdiagnosed monogenic diseases across African health system?
Right. Well, obviously, this is a very good question, and actually, I fully agree. I mean, and we work on that. We clearly work on monogenic disorders. I mean, the most common monogenic disorders of Africa is sickle cell disease, right? I mean, and this is easy because this is a very small gene, the HBB gene, and we have been demonstrating that we have a workflow for HBB that is very efficient. Within a couple of hours, you can get. Well, we did proof of concept tests in Dakar, and you can get within a couple of hours the full perfect sequence of patients where you suspect to have variants in the HBB gene.
Okay,
But
Doctor.
I'd like to just follow up. Sorry, I was distracted by something else. But HBB is easy. We have two papers that are now in press or published. One is Wilson disease, which is this transporter of copper that also recessive disorder. And again, in a small cohort of 12, or I think it was 12 patients from Morocco, we found new variants. And again, here, I didn't stress that, but one of the big advantages of sequencing the whole gene is that you don't have, you're not restricted by panels. So because you have the whole exons and introns, so you can get intronic also variants, right? And we found intronic variants, for instance, in Wilson disease. And these intronic variants would never be seen if you do panels, or sometimes it's very difficult to find them also, some of these structural variants if you do short read sequencing. So we have been working on CFTR, HBB, ATP-B7 for Wilson disease. We now are working with Tunisian colleagues with SCN5A, which is a gene coding for a cardiac arrhythmia gene. And we also have we demonstrated it works well for BRCA1, BRCA2 for breast cancer.
Okay, thank you, Doctor. We have David here. Please unmute yourself and ask your question.
Thank you. Good afternoon. And thank you very much for the lecture. It's been an insightful session, and the questions have especially been helpful too. Forgive me if I missed this during your presentation because I did a bit of a spotty connection earlier. But so far, the first question is, what's the landscape of the mutations that you have been seeing in your work with CFT? I'm curious because, like you mentioned, those medications, and they've revolutionized the.
So for the moment, most of the mutations are missense mutation that we observed in these patients. Yes, and then again, many of them were already. I mean, and we did that for not that many patients from Morocco, and many of them were already known as being pathogenic variants, but others are clearly VUS. We didn't find so far big rearrangements or structural variants. We didn't find neither anything that would be intronic for CFTR. We found intronic variants in BRCA1 and in ATP-B7. Actually, these intronic variants, 2 times we got, in fact, ALU insertions in the introns of these genes that we think would have been pathogenic. Is this answering your question, David?
I think his
David, you are on hold.
connection is
Yes.
not good.
Yes, he is on hold.
Okay.
There is another question, Doctor. What
He
is the
back.
realistic?
He is back again, David.
Okay.
Yes, sorry. I don't know if you can hear me clearly now.
I hear you, I hear you well. Yes, yes, David, go ahead.
Oh, great. Yeah, so I was asking from your research so far, what's the landscape of the mutations that you're seeing? Are they going to be compatible with, those medications we've talked about, or are we seeing variants that are leading to non-functional
Okay.
proteins that can be helped with that?
Right. That's a very good question indeed. Both. Yes, and this is why indeed functional testing I mean that's obviously something, an added value of our lab. We can do some sort of functional testing, and in some functional testing we observed rescuing, and in others we didn't observe rescuing, and this is clearly the basis of precision medicine, right? So I see one question. I'm reading a question here.
Yes,
What's
Dr.
the realistic perceivable cost of nanopore-based CFTR sequencing in West Africa? Obviously, we didn't perform a cost analysis, but what I can tell you first, the investment to buy the device. Now there is a new device. You have a laptop plus a smaller device. It is about 8,000 to 10,000 euros, right? So that's the buying the device. So you can start to sequence with less than 10,000 euros, and the results that we get, we interpret these results as diagnostic ready. I mean, clinical grade results. We didn't do the performance, all these things, but we think that the quality of what we're observing is very good. I mean, I would really trust to give these results to the patient. Then, you have the consumables, and then I would say, a really rough estimate per patient, even though you can multiplex. I mean, Nada, when she was doing this cohort of patients from Morocco, she had no problem to put 20 patients in one sequencing cell with barcoding. So I would say, it goes from 50 to 100 euros. That would be the range I would estimate.
Okay, Doctor, we have...
That was answering the question of, I think,
Osman.
Yeah.
Yes. And here, Dr. Wafa, I have another question saying many students attending today may be interested in postgraduate studies. What qualities do you look for when you select master's and PhD candidates for your laboratory?
Okay. So there is one way to get into, my lab or another lab in Switzerland. Switzerland has scholarships for excellent students. Every year, you can apply to Swiss embassies for these excellent scholarships. Nada from Morocco got that scholarship, right? She had to write her project. She had to, defend her project with me. I had to support her project, and then she has been selected. Okay. I had an Egyptian colleague who came. He was from the University of Zagazig, from Egypt, a pharmacist. And again, that was similar. So he contacted us. We wrote a project together, and he got the money so that he could do his PhD in our lab. I mean, I think in terms of qualities, again here, one of the qualities that we could that would come from what I just mentioned is independence, right? You have to have been already a clear, independent mind, and you have to know what the PI in the lab would expect from you. That there is a clear exchange with the PI.
I think, Doctor, you gave us hope, and I already appreciate this answer, actually. Okay, if anyone wants to ask anything more, please feel free and raise your hands.
What I'm going to do, I'm going to put in the chat
Okay.
the link for the scholarships.
Okay, that's good.
Yes, here. Right. So those are Swiss Government Excellence Scholarships for PhD students. And again, I mean, and I just had a young colleague from Congo, from Bukavu, where there's war. He just got one, and he will be in Switzerland in about 6 weeks.
Congratulations. Congratulations.
If there are no more questions, are we going to stop here?
Yes, I think we don't have other questions.
There is
we
one
have
here.
one. We have one, yes. Your Moroccan study identified novel CFTR variants. How should clinicians and researchers interpret variants of uncertain significance in African populations?
Right. That's a very good question, because actually, if you don't have access to, I mean, well, the first thing is that you have to go to databases and see whether there is something in ClinVar or whether there is something in the CFTR database of pathogenic variant. If it is not, so you have to assume it's a VUS, obviously. One of the things is that you would have one thing that would be nice. And this is something that has started in many African countries. You start to have your own genomes of reference, right? I know Egypt has started to do that. Morocco has started to do that. Senegal wants to do it. And many, many countries in Africa want to have their own genome of reference. So, and then you should have, with the genome of reference, you should also have the allele frequency of variants, right? If the variant that you find in the CFTR gene is frequent, let's say more than 1% or more, something like this, so it's most likely not a pathogenic variant. I think it stays a VUS. Then I'm not. I mean, obviously, trying to treat the patients with these drugs is almost impossible, because these drugs are extremely expensive. Then the other things would be, to team up with labs elsewhere in the world that would look at functional assay that would demonstrate a possible pathogenicity of these variants.
Okay, doctors, thank you again, Dr. Hugues Abriel, for answering the questions and for your excellent presentation. It was a pleasure to have you with us today. Thank you all for attending. We hope you found your informative and enjoyable. Have a great day, and thank you so much for attending.
Thanks a lot. Good luck to everyone.
Thank you, Doctor.
Thank you so much, Dr. Abriel, for your time and for your informative presentation. Thank you so much. Have a great day. Bye-bye.